So in these set of videos, we're going to be talking about blood fluid contents, and this information comes from Guyton chapters 33, 34, and 37. The presentation doesn't really organize the information by chapters, more by topic, which we'll see in just a second. So this is kind of a where we're going overview. So we're going to talk about red blood cells, white blood cells, platelets, and endogenous anticoagulants. These are the main components of the blood, and so this is why we're organizing the presentation by topic rather than by book chapter. So jumping right into red blood cells and their structure. So when we think about red blood cells, we think about the typical biconcave disc. We've probably seen lots of images of this in textbooks. But what's important to note is that there's a lot of excess cell membrane for red blood cells, which allows them to be compressible and change shape. And this allows them to undergo diapedesis, or their ability to squeeze through the endothelium and exit the blood flow into the interstitial space. But when they're free floating in the blood, they do have that biconcave disc shape, and that shape actually maximizes the efficiency of their flow. So red blood cell production starts, like most cells, with an undifferentiated progenitor stem cell, but the first differentiated cell is the proerythroblast, and it'll undergo all of the steps you see on the left to eventually form erythrocytes, which are red blood cells. Production changes location throughout development of the fetus. So in an early embryo, red blood cell production occurs in the yolk sac. In the middle trimester, it moves mainly to the liver and also still in the yolk sac. And then in the last 4 weeks of gestation, production of red blood cells almost completely goes to the bone marrow. And at first, it's going to be in all bones, the bone marrow of all bones. By the time you're 20 years old, it'll actually be only your membranous bones, so your vertebrae, your sternum, ribs, and ilia. Two important factors to think about when we're talking about the production of red blood cells is vitamin B12 and folic acid. And the reason why these are important is because a deficiency in either of these can lead to something called megaloblastic anemia. So vitamin B12 and folic acid both are essential for DNA synthesis. And without them, you result in a red blood cell maturation failure, which forms these types of red blood cells called megaloblasts, which are very large with very thin and fragile cell membranes that are ruptured really easily. So vitamin B12, the liver actually stores 3 to 4 years worth of B12. So the only way you're going to have a deficiency that's really significant that can cause anemia is if you are deficient in B12 for a very long time. And something that can cause that deficiency is called pernicious anemia, and that's when you have a low gastric secretion of intrinsic factor, which intrinsic factor is the component that helps B12 be absorbed. So without it, you can have anemia. And then folic acid is the same way. If you're deficient in folic acid, you get the red blood cell maturation failure. And a disease that can cause this is called sprue. It's a small intestinal disease that affects the absorption of folic acid. And we'll talk about megaloblastic anemia in more depth in the video specifically relating to pathology. So red blood cell production is actually regulated by erythropoietin, and this is an important connection point between the renal system and the cardiovascular system. So erythropoietin is produced by the kidneys mainly, about 80%, and then 20% by the liver. And so when you have hypoxic renal tissue, that can induce the expression of hypoxia-inducible factor 1, and that factor stimulates the production of erythropoietin in the kidney. And so in your tissues, if you have erythropoietin and you have hypoxia at the same time, that will stimulate proerythroblast production, as well as quicken maturation of those into erythrocytes. So basically, you'll get a lot of red blood cells really fast. And then once you're sufficiently oxygenated in the tissue that was having the issue, erythropoietin returns down to maintenance levels. And all of this is explained really clearly in the image to the left. And it also includes some of the factors that can cause low oxygenation. So low blood volume, anemia, low hemoglobin, so you're not carrying enough oxygen, poor blood flow or pulmonary disease, so you're not getting oxygen in the body at all, or at least not enough. So there's two important components inside of red blood cells that we need to talk about. The first is hemoglobin. So hemoglobin is actually the portion of the red blood cell that carries oxygen. And so one hemoglobin molecule is made out of 4 hemoglobin chains. And what you see on the left is a heme moiety. So in order to make a hemoglobin chain, you take one heme moiety and you add a globin polypeptide chain. And so the heme moiety you see on the left is centered around iron, and iron is actually what binds to molecular oxygen. So every heme moiety has an iron center that binds to 1 molecular oxygen. Therefore, every hemoglobin molecule can bind to 4 molecular oxygen. And so the iron is really what's important with hemoglobin because it is what binds oxygen. It is what allows us to carry oxygen throughout the body. And so where does the iron come from? So iron is absorbed by the small intestine through your diet. And once it is absorbed into the plasma, it binds to a protein called transferrin, which you can think of as the transport protein for iron. And transferrin's job is to bind iron and take it to the red blood cells for it to be packaged up into hemoglobin. And we lose iron in a couple of different ways. We can lose iron through blood loss. We can lose iron through feces excretion. And then also when your red blood cells are damaged or are old and dying, they're not working properly, macrophages actually come in and degrade that red blood cell and degrade hemoglobin to release free iron. And so free iron, there's a couple of things that can happen with free iron. It can either bind to transferrin again and be taken to make more hemoglobin. It can bind to a protein called ferritin, which is a short-term, smaller storage place for iron. If there's a lot of iron, it can be bound by hemosiderin, which is more of a long-term, it's a really large deposit of free iron. And then iron again can be packaged to create heme, or it can be degraded by enzymes. So the second important factor that's in red blood cells we need to talk about is the enzyme carbonic anhydrase, which we've talked about in class a little bit already. So carbonic anhydrase is the enzyme that converts CO2 and H2O into carbonic acid, and it can also do the reverse reaction as well. And then once you have carbonic acid, it can dissociate into bicarbonate and hydrogen ions in water, or in the presence of water. So this is actually the way that CO2 is carried in our blood until it can get to the lungs, because if you think about it, if you could only carry oxygen carbon dioxide as dissolved gas, as you went along the system and we're collecting more and more carbon dioxide from the tissues, at a certain point, you would have more carbon dioxide in the blood than in the surrounding tissue. And so that carbon dioxide gas would no longer be able to diffuse into the blood. So we have to have some mechanism to convert the CO2 to something else in order to continue to pull in more CO2. And the way that happens is CO2 is converted into carbonic acid or bicarbonate. And the other important thing to note about carbonic anhydrase is that the carbonic acid and bicarbonate act as a buffer system for the blood. So carbonic acid is a weak acid, bicarbonate is a weak base, and so having the two of them allow you to fluctuate the pH of the blood a little bit. So if something happens to where the pH changes, we can really easily be able to alter the pH of the blood to try to maintain homeostasis. And this is kind of an image that summarizes everything we've talked about in this section for red blood cells with the hemoglobin, the carbonic anhydrase, and the regulation by erythropoietin. So this is it for the section on red blood cells. The next video we'll talk about white blood cells.